as we get ready, perhaps each of you can just state where you are and your affiliation and title. I think we know all of you, but just for the few who might not have met you.
Mark Blumenkranz. I'm a professor of ophthalmology at Stanford. I'm also a serial entrepreneur in the biomedical device and drug space.
My name is Don D'Amico. I'm professor and chair at Weill Cornell, New York Presbyterian, and I'm up here to give Mark all the answers.
I'm Pravin Dugel. I'm a retina surgeon in Phoenix, Arizona, and a clinical professor at the USC School of Medicine.
Peter Kaiser. I'm a retina specialist at the Cole Eye Institute. Do a lot with reading centers for clinical trials.
So Adnan, Biotech Analyst at RBC with the Wall Street perspective.
Great. So thank you. These, for those of you, most of you know them. They're very seasoned and experienced folks in retina, so it's good to have you here. The first question we have for you is the therapeutic promise of targets and the proposed mechanism of actions just presented. Did you hear anything that really excited you? I'm sure you've heard of these things before. And can you comment on other promising approaches in terms of combination therapy to the current existing anti-VEGF? So, Pravin, I know you've been dabbling in this area for some time, so why don't you comment a little bit about what mechanisms, in addition to anti-VEGFs, do you think are important?
Well, Mark, I think this is a really exciting time for us.
Pull it a little closer, Pravin.
Yeah, I think it's a really exciting time for us. I think we're in a position from a clinical point of view as well as from an industry point of view where I think we realize that as good as VEGF-A suppression is, I think that we've reached a ceiling effect of that, and we're starving for combination therapy. So I think this is a really good time. I'm excited about all the approaches. I think the strategy is kind of interesting, though, which is that there are several great companies going after a particular mechanism of action. You know, Ang and Tai are the ones that come to mind, which validates that target because they're great companies going after that. There are other companies like Opthea that are going after validated targets, and they're the only ones that are there, such as pan-VEGF inhibition. And there's an advantage to both. I think if you're the first one there in a mechanism of action that's unique but validated, I think you potentially gain a dominant position. So I think that's an exciting thing on both ends, on both strategies. And finally, there's a delivery system that's also different. I think I'm excited about the Arpaio delivery system and the fact that there's a whole new door that opens up with the potential treatment for diabetic retinopathy. And it would seem to me that that delivery is ideal for diabetic retinopathy, being sub-Q and also going to both eyes. So I think we're in for really exciting times, but we're starved for combination treatment, and I think we're in the precipice of changing our treatment paradigm.
I have 3 or 4 thoughts. I think you can sort of group the improvements into getting compounds that have slightly greater intrinsic efficacy or getting better delivery, or you can look at new targets, and we heard today about TIE2 and a number of other things. But I would say that there's a lot out there that we could do that we probably aren't doing as effectively or efficiently as we could be, and one of it is, as opposed to combination therapy, I think multimodality therapy is either a subset or a superset of combination therapy. For instance, that might be laser plus drugs, it might be surgery plus it might be intravitreal injections plus gene therapy. So I think mixing and matching those things works well. Another thing is looking at naturally occurring agents that are more non-specific. I mean, steroids are a great example. We produce steroids, and yet we still rely upon formulating steroids, and they have a great effect. There's another class of compounds called crystallins, alpha and beta crystallins. We produce those. They're close to a super family called heat shock proteins, which are some of which are chaperones, some of which are solubility factors, some of which are neuroprotective factors. And if you think about it, it's really interesting when we use low-dose laser, whether it's micropulsing or whether it's very low-fluence laser, it looks as though that effect, which may be better than we had ever hoped for, is actually modulated by the expression of HSP70 and other proteins. And some of these are involved in really complex things like folding, tertiary folding of proteins. So that's another area. And then the third thing I'll mention is, when you think about it, it takes 2 to tango. When you have essentially a disease that's mediated by a cytokine or an aberrant protein, it takes a receptor to modulate that effect. These are not toxins, they're cell modulators. And almost 100% of the time, up to this point at least, we've focused on mopping up the agonist or the ligand, and whether it's a monoclonal antibody, or which is not naturally occurring, or whether it's something along the lines of a trap, which is the fusion protein with portions of the receptor. Nobody's really looking at binding the receptor, and if you bind the receptor, you can actually inhibit in a non-stoichiometric way. In other words, you can only bind as much agonist as you have binding protein, but if you block a receptor, you can produce an infinite amount of agonist or ligand, and nothing much happens. Now, that's probably overly simplistic because you have to be sure that on the one hand you're binding the receptor and not turning it on, but there are ways to do that using a structurally modified protein. So I'd say those are the areas that I think are really promising and that I think have been investigated up to this point in time.
Don?
So I thought the presentations were incredibly interesting. I think the Tie-2 Ange story is really the one that, to me, strikes me as the closest to making sense, having data, having mechanism that really looks like there's a drug in there somewhere. I'm very interested in, but a little bit confused by, the integrin approach because it seems to do a lot of great things, but it does two wildly different things. I mean, it changes your hair color and takes your shoes off, so to speak. And it's how
wrong with
those will
that?
play in the diseases that we're treating. Some diabetics, we've learned that inducing a PVD is a great thing. Years ago, people tried to do it with ultrasound and other things to try to prevent progression of the disease, as you guys well know. But I'm not sure if I right away want a mechanical effect with a biologic effect. So, but they've treated zillions of over 1,000 injections. So I'm excited about that. And finally, the approach that I really still think we have shortchanged steroids. We've got a couple of generic and now approved triamcinolone preparations. We have fluocinolone implants and Dex implants, and sort of that's it. But yet, I think all the panelists would say if we could recall and bottle in a safe way that wow effect that we got in some of these diseases with the initial intravitreal triamcinolone work, we would really want that in our armamentarium. And yet, it's too much like a baseball bat. So I would hope that we could continue our work of understanding precisely how do steroids produce that effect, and not just giving the class of compound that we found available on our shelves.
Peter, go ahead.
So, we've been concentrating a lot on combining and making all these diseases one disease that we can treat the same way. But if you really think about it, macular degeneration, diabetic macular edema, even RVO, they're very different diseases. It just happens to be that anti-VEGF works in all of them because it's a leakage problem. But if you look at macular degeneration, we've reached a plateau in vision. We're not really causing the CNV to disappear. We're decreasing leakage, and that's where all these other pathways come in. If you look at the angiogenic cascade, every single thing we talked about today is within that angiogenic cascade, and which combination is going to work remains to be seen. But what works in AMD may not be the same thing that works in DME. So for instance, the Tie-2 pathway, very important in diabetes, probably a little less important in AMD, but that doesn't mean the drug shouldn't go forward. I think in the future we'll be more talking about these are the DME treatments, these are the AMD treatments.
Right. I think that brings us to the next slide, I believe, about the different mechanisms, if we have that in order. Emmett, I think you're...
I have the controller, sorry. So just sort of to generalize that away from the specific targets that we've talked about and more toward what these signaling pathways are doing. Are they neuroprotecting? Are they causing fibrosis, permeability, angiogenesis? Adnan, maybe I'll switch back to you and to step away from the assets we heard about. When you think biologically, what do you as the analyst, as the scientific mind behind all this, which of the sort of targets and pathways are most attractive to you in AMD versus DME?
So Emmett, if I may, I'll take that a level higher. So the question I'll answer is what kind of companies can we fund on the street? What kind of companies are of interest to Wall Street? At this time, both companies that improve vision, improve a clinical benefit, as well as those that improve duration, they're both fundable companies on Wall Street. So we haven't really changed the story that much from 2-3 years ago. Yes, new mechanisms are fantastic, and they're fantastic because all of us on the sell side and even on the buy side are looking to run from Avastin as being the reference drug. So whatever you can give to us and explain to us that will no longer put Avastin as a competitor, that is a fundable asset.
Okay. Anybody want to speak to the target science? And are these, let's stick with AMD first. Is AMD more about dying cells, fibrosis, or persistent fluid/vessels?
Well, I think we know how patients lose vision. I think we know that over 50% of patients end up having scarring if you follow them long enough. I think we know that in all cases of anti-VEGF-A monotherapy, there's a period of time after which patients start losing vision gradually. And I hear what you're saying in terms of the importance of vision, and I truly get that. But the drugs that we have right now are really good drugs. And at the end of the day, I think when we develop drugs, I think right now we have a biomarker crisis, because I think history will show that it's an absolute coincidence that we've developed an anti-permeability drug or drugs, VEGF-A suppression, at the same time that OCT developed. At this point, how do we show, other than vision, and the vision gain, the margin is really small in terms of what biomarker do we use for fibrosis, what biomarker do we use for vascular stabilization. And I think that's the biggest challenge that we have in drug development, is that we simply don't have a good biomarker for these things as we did, and we've been spoiled by the biomarker that we have for permeability.
Done.
I'd say that from this list, anti-fibrosis, I think is my favorite. The neuroprotection one has been incredibly hard to pin down, even in something as simple, for example, as a rhegmatogenous retinal detachment. Still, for all the years that's 100 years now, it's been successfully treated, there's still nothing that we do other than put the retina back into its native position that helps the retinal function. We haven't figured out, should we inject glucose in the vitreous, or should we hyperoxygenate people? I mean, Dave Zaks and others are looking at adjuvant compounds. So the neuroprotection one is going to be very, very hard to prove. I think anti-permeability and anti-anginesis are important, but anti-fibrosis, if you could preserve a retina in a reversible state and not have it become a scar, I think you'd live to fight another day. So I would put that at the top of the list.
I want to ask Adnan, and then I'll let you comment, Peter, just to follow up. We didn't list here durability, enhanced durability. So from the street's perspective for AMD and DME, what is the, okay, I'm interested? Is it 3 months, every 3 months? Is it 6 months? What do these companies need to get to as far as dose interval for the street to say, now you have me?
So 6 months would be fantastic, but 3 to 4 months is acceptable. And as you know, there are phase 3 studies ongoing with that target in mind.
Okay, Peter, I'm sorry, I didn't mean to interrupt you.
No, that's all right. I mean, I think if you look at macular degeneration in particular, none of our drugs are anti-angiogenic. And that's the big problem. Maybe that's why they become fibrotic, is because we're really not reversing CNV. If you look at any of the papers, Marina, Anchor, View, even some of the later Ophthalmology, the CNV is not disappearing, and that's the issue. We need to really cause the CNV to disappear. We don't have anything that does that yet.
And I think
Don,
question
and we'll
for
go to the
Adnan.
next question.
I was just going to comment on that, Peter. I think that's the holy grail. It's very difficult to have those blood vessels disappear and yet not cause fibrosis. So how do you do that? I mean, how do you get these blood vessels to disappear and not cause fibrosis is really the interesting thing. I think the other that was perhaps left off is inflammation. We didn't really talk about that. And I think if you blunt the inflammation during this process, you get a little bit potentially less fibrosis as you're taking away that blood vessel. But Don, you had
I
something?
had a question for Adnan. So Adnan, if there was a therapy that came out that was not quite as good as first-line therapies for AMD, but was given at every 4 months, but not quite as good, how close would it have to be? For example, in the antifungal world, when they invented fluconazole compared to amphotericin, I think the figure in the New England Journal trial was it had to be like 75% as effective because it was so simple to give orally. So is there an analytical point at which a sort of a long-lasting would be marketable even if it wasn't quite as good as monthly injection?
So again, I'd go back to 2 points. First, it's the Avastin being the reference drug as the bugaboo. So maybe you get it used, but it doesn't quite get used frontline. But and you know, all of us on the street, because there is combination data coming by the end of the year, both for Vista and the Eylea plus PDGFR drug, we're trying to determine what is that meaningful letter difference. And frankly, probably we're sort of on the same page as the ASRS survey shows, that 4 or 5 letter, 1 line improvement is something meaningful. But where the buy side and sell side are, I think, focusing is something Dr. Boyer pointed to in his presentation, that 3-line gainers, 4-line gainers, maybe there are subsets of data that you can look at which become meaningful from an efficacy standpoint. So when you doctors come back to us and tell us something is meaningful to you, it becomes meaningful to us as well.
Go ahead, Mark.
I think I have a slightly different view than the others have expressed. I think the difference between 1 month and 2 months and 3 months and 6 months are primarily tactical advantages that you have over the short term, because there's always a leapfrog possible. I think what people really are looking for is to cure these diseases, and probably the best way to cure disease is to prevent it. And the second best way is to have an effective treatment. So, and there are ways to cure these diseases right now. We have the technology to do this, whether it's protein supplementation using things like gene therapy, or whether it's gene editing. Now, we don't have the luxury of gene editing some of these diseases because they're polygenic and it's just too complicated and there's too much work to be done. But we do have within a reasonable sight line the ability to supply proteins at an early stage in these diseases. We know from experimental models, a number of us have been in the lab for years, it's much harder to cause regression of an established disease, such as myocardial contractility or stroke, than it is to prevent it. The catch, the bogey is that you have to know who's going to get it, and then you have to have a therapy that's safe and effective and relatively non-invasive to do that. But if you could do that, we'd all have that. I'd have that, I'd have that treatment. So I think we have to look a little harder into not treating these diseases, but preventing these diseases, and I think it's within our reach to do that right now.
Okay, let's, was it a burning comment or quickly, or do we move on?
Go ahead, Pravin.
A quick comment. I mean, it's not clear to me, as we understand the physiology of neovascularization, that getting rid of the blood vessel is actually the thing we want to achieve. It's a wound healing response, and I look at it more as modulating the blood vessel. I think there's a reason the blood vessel is there in a wound healing response, but we're just hearing about it. And finally, the other comment that I had is when people ask me all the time, what's a significant improvement, 4, 5, 2, 3 letters? That's the median letter gain that you get. It's a very, very widely distributed disease. The next question ought to be, what's the distribution? If there are patients if the median is 4, there's a subgroup of patients that are doing really well if you can identify that subgroup. At the end of the day, the question really should be, do you think this study will hit its primary endpoint in registration? And if it will, it absolutely will be used because our threshold is so low.
Mm-hmm.
So we've touched on this question a bit. I'll let you take the next one if you want, and
Okay.
since we've touched on this one. I'm not sure if we want to hit on any other points. This was intended to make the point that we're going to be seeing this data come out, 4 or 5 line mean gain or change, injection burden. We've talked about how much is really desired. Anybody want to make 1 or more comments about how to interpret these data as they come across our desks, whether it's 4.5 letter mean gain or?
Well, I think Pravin hit it on the head. 4 to 5 letters, it's a mean. What matters is are you like Alex Rodriguez? So you either hit a home run or you strike out, or you're more like Ichiro and you get on base all the time. And which one of those two you are will matter. So personally, I'd rather see a drug that has a mean where you have a whole bunch of super winners, and very quickly you can see if you're not a winner and switch them to something else, versus just a minor improvement in vision.
Okay, so let's go on to the next question, and perhaps maybe the last question is, assuming approval of 1 or more of these discussed drugs as an add-on and adjuvant therapy, how will this impact your patient flow practice? I mean, what do you think, how will this play out? Will you be doing 2 injections? On the same day, would you inject and then perhaps treat with the other drug periodically? How do you see it, Doc?
So I have some views on this. I'm not a fan initially of immediately going to combination drugs, because I think we've only had sort of 1 class of agents, maybe 2 if you count steroids, and now we're going to combine them. and we'll get into the same problems we got into with all the combined glaucoma drugs. I would like to be able to use them individually as we explore. I also think the injections are not going to be a big deal. It often takes me less than 3 or 4 minutes between the time I tell a patient that they're going to get an injection and then they're out the door. And I think we're going to go bing, bing, and I'm not worried about the volume. I think that as the volume increases, as it certainly must, we will have ways to deal with it, whether we get, and I don't think it's going to be multiple visits. I think you're going to give and then very quickly give a second injection. The 30 minutes is going to be reduced quickly in clinical practice to just a few minutes. That's my, I think it's going to be a non-problem, I guess is what I'm saying.
Okay. Peter, what?
Yeah, I would agree. I think an injection or 2 injections, it's not going to matter. Patients will do it if it improves their vision. That's the bottom line. Vision trumps all. So if it's injections, no matter what, we'll do it on the same day. We're not going to change how we do it. The thing that's going to be interesting is if, for instance, the sub-Q injection of AirPO works. That's something I don't have to worry about in my clinic, which is great. If the OR eye drop works and I can give them a prescription and go just take this eye drop, that would be great. So there is going to be some differences in the future, but again, what we'll use won't matter except for the vision. So whatever's the best vision, that's the drug we're going to use.
Pravin?
I think at the end of the day, efficacy is king. Efficacy always wins. And I'm old enough to remember when we sat there and said, who would ever take a needle in their eye once every 4 or 5 months? And look at what we're doing now. That's number one. Number two is I really look forward to having a portfolio of agents. And the beauty of this is that none of these mechanisms of action are exclusive to another. I hope they all work. And I hope we'll use them all. And I look forward to having a portfolio where we'll be able to truly individualize and customize care. And I think that's what it will come down to, the practice in 2020 where we'll see patients and we'll be able to individualize treatment and customize their selection of combination therapy. I think that's the ultimate goal.
Great.
Thank you all. We've run a bit late, and I have to thank my panelists. Thank you for initiating this long and complicated discussion.